I'm looking at a section of rat pancreas at a magnification of about ×400. I want to find out all I can about the structure of the pancreas and about its function. This slide has been prepared by chemically fixing a piece of pancreas which has been embedded in paraffin wax. A thin section has been cut, which has then been stained with hematoxylin and eosin. Eosin, an acid dye, stains the cytoplasm red, whilst hematoxylin, a basic dye, stains the nuclei blue. Examination of the tissue reveals that there are two different cell types in the pancreas. The islets of Langerhans are these lighter coloured clumps of cells, which occur throughout the pancreas and occupy about 2% of its volume. The rest of the tissue contains the acinar cells, and it's these that are believed to be responsible for producing enzymes. But how would we show that the pancreas is responsible for producing enzymes? We know that the pancreatic duct links the pancreas with the small intestine, and if we cannulate the pancreatic duct, that is, if we attach a thin tube to the pancreatic duct and collect the secretions that are produced, we can find that this contains enzymes. At higher magnification, it can be seen that the acinar cells are arranged in a cluster around a central duct. But this is about the limit of resolution from an optical microscope. One of the crucial factors in determining resolving power is the wavelength of the illumination. In general, the shorter the wavelength, the greater is the resolution. The wavelength of visible light does not allow us to obtain magnifications much greater than about 1500 times. However, electrons have a much shorter wavelength, and if we use electrons as the illuminating source in an electron microscope, we can obtain much higher resolution and therefore much greater magnifications. This glass slide is not, however, much use in an electron microscope, so I'll have to start with a fresh piece of pancreas. A small piece of tissue is removed from a rat and placed on a wax block. In electron microscopy today, a technique of double fixation is used. The first fixative is glutaraldehyde. This penetrates tissue very slowly, so small pieces of pancreas are cut, ideally about 1 millimeter square. This enables the fixative to penetrate all parts of the tissue. Glutaraldehyde contains two free aldehyde groups, which cross-link with amino groups in proteins, and so fixes these in the tissue. Prior to using the second fixative, the tissue is washed in buffer to remove excess glutaraldehyde. Osmium tetroxide is then used primarily to fix lipids. The fixed tissue is then passed through alcohols of increasing concentration. Once dehydrated, it's transferred to resin. It's then put into a small mould and placed in an oven to polymerize. The viscous resin hardens and the tissue, the black material in the resin, can now be sectioned. Whereas in optical microscopy, sections about 10 microns thick are good enough, in electron microscopy, they need to be 200 times thinner, as electrons have such a low penetrating power, and these sections are cut on an ultramicrotome. By careful alignment of a sharp glass knife edge with the specimen, ultra-thin sections can be cut. the specimen holder moves down in the knife edge, producing a ribbon of sections some 50 nanometers in thickness. Copper grids are used to support the specimen. We can't use glass because electrons have too low a penetrating power. They can't get through. The specimen is then stained. Firstly, in uranyl acetate solution, the uranium salts selectively bind to proteins and nucleic acids. Staining with a second compound, lead citrate, makes use of the ability of the lead salts to selectively bind to lipids. By this double staining technique, the contrast of cell components and membranes is greatly enhanced. The electron microscope looks very complicated, but in principle it's really quite simple. The source of illumination is the electron gun at the top of the column, and when a voltage is applied to the electron gun, a beam of electrons is emitted which passes down the column, and the column's under vacuum because electrons don't travel very easily in air. The electrons are focused by a series of electromagnetic lenses, which are analogous to the glass lenses that we have in a light microscope. The electrons are focused by the condenser lens onto the specimen, which is here. And the image of the specimen is focused by the objective lenses and magnified by the projector lenses. But there's a problem. Electrons can't be seen by the naked eye. So in the electron microscope, we have a screen which is here, and the screen contains phosphor crystals. When electrons hit the crystals, they fluoresce and produce the image which we see. The grid has been placed on this specimen holder, and I'm now going to put it into the microscope. I can't put the specimen directly into the microscope column because you remember that is under vacuum. So first I put it into this airlock. And this is it, a real live image of the section at a magnification of about 4000. But to the untrained eye, it's a bit disappointing. It's hard to make out what things are. At the bottom, the light area is actually connective tissue, and above this is the triangular-shaped pancreatic acinar cell. This is the nucleus. These could be secretory granules. But this type of image is difficult to interpret. It's produced by phosphorescence in the television tube, and it lacks clarity. However, attached to the electron microscope is an automatic camera, which takes black and white photographs, and these produce the more usual image. This photograph, in fact, has been taken at a similar magnification, but just look at the difference in clarity. But if we need to look at the cell in more detail, we can't just increase the magnification of this photograph. If we did, we would lose resolution. So we must increase magnification on the electron microscope itself. However, this gives us a large number of photographs. But by carefully piecing these together, like the pieces of a jigsaw puzzle, we can build up a composite picture of the cell. This is called a montage. Here I have a number of photographs taken at a magnification of about 44,000. And I'm piecing these photographs together in the region of the nucleus. This fits quite well together. I'm just matching up the nuclear envelope. Here is a completed montage of the same pancreatic acinar cell. This is the basal membrane, and beneath it, the light area, is the area which contains blood vessels and connective tissue. This dark structure is probably a red blood cell. Here is the basal membrane again, and what appears to be an extensive, rough endoplasmic reticulum. If we go in and look at this at higher magnification, you can see a number of dark blobs along the surface. These are ribosomes, which indicates that this cell has got a considerable synthetic capacity. As we move up the cell, here is a mitochondrion, which is a number of transverse cristae. And here is the nuclear envelope. We call it a nuclear envelope because it consists of two membranes. On the outer surface are a number of ribosomes, and on the inner surface is condensed chromatin. As we move up the nucleus, there is a large nucleolus. And when we move on up the cell, towards the top of the nuclear envelope, there is more rough endoplasmic reticulum, mitochondrion, and here what could be a secretory granule. This would contain enzymes. And here is the Golgi apparatus. If we look at this in more detail, we can see sacs of what are called Golgi cisternae. And at one end, we can see a number of what are probably condensing granules which are being packaged by the Golgi body. As we move up the apex towards the apex of the cell, there are many more of these large granules, and these almost certainly contain enzymes which would be secreted within this acinar cell. These large granules are bounded by a membrane. It's difficult to see at this magnification, and as we move up towards the apical membrane of the cell, we can see that there is a lot of floccular material in what's the lumen at the top of the cell. These large granules, the large dark granules in the cell, are probably secreted into the lumen. So the enzymes would be secreted from the cell into the lumen and passed down to the pancreatic duct. So electron microscopy has shown that the pancreatic acinar cell, with its extensive rough endoplasmic reticulum, has a considerable synthetic capability. But if we want to know more about the movement and synthesis of enzymes within the cell, we need kinetic information. We need to be able to follow the movement of materials through the cell. And for this, we use a special technique called autoradiography. In vitro, pancreatic acinar cells are exposed for a short time to amino acids containing a radioactive label. This is called the pulse. The cells are then transferred to a flask containing unlabeled amino acid. This is called the chase. So a short pulse of radioactive amino acids will pass into the cell. After fixing and sectioning the pancreatic tissue, a photographic emulsion is spread over the grid and stuck in this glass slide for ease of handling. When radioactive particles interact with emulsion, silver grains are deposited, and it's these which indicate the position of the label. The longer the period of chase, the further will label have moved through the cell. Dr. George Palladdy from Yale University pioneered this work.
The preparations with the autoradiographic grains on them are examined in the electron microscope. And what we see here is the situation found at the end of the pulse. The autoradiographic grains are over the endoplasmic reticulum. After 10 minutes of chase, the grains move from the endoplasmic reticulum to the periphery of the Golgi complex. After 40 minutes of chase, they are on the other side of the Golgi complex over condensing vacuoles. And after an hour or 2 hours, they are over mature six-story granules, which are called, in this case, zymogen granules. To quantitate the results, we count the grains over each compartment as a function of time from the end of the pulse during the chase. At the end of the pulse, the majority of the grains, as indicated on this graph, are on the rough endoplasmic reticulum, but they move very fast out of this compartment. At 60 minutes during the chase, they are over the Golgi complex. At 120 minutes, they are over immature six-story granules. And later on, they accumulate progressively in mature six-story granules. The results are very clear when the protein for secretion is secreted at a higher rate than the protein for intracellular use. In the case of the pancreas, the difference in rate is from 10 to 20 times. Therefore, 1 grain in 10 will represent the background. 9 grain will represent six-story proteins.
Another way of looking at specimens for the electron microscope is to freeze fracture them or freeze etch them. This is particularly useful for looking at cell membranes, and it involves freezing the specimen and then cracking it open, more or less like cracking a block of ice with an ice pick. To freeze etch a sample, the first thing you do is put the sample on a small copper planchette, which you then plunge into liquid freon. After the specimen is cooled, it's placed in a storage dewar so that it can be carried to the freeze etch machine, where it's put on the specimen stage. Inside the freeze etch machine, there's a specimen stage, and poised just behind it, the knife, which is used to cut the specimen, and focused on the specimen, just above it, are 2 electron bombardment guns used to cast a replica of the specimen after it's been fractured. Once the specimen is inside the freeze etch machine, the bell jar is lowered, evacuated, and then the knife's used to cut the specimen itself. Thin slivers are shaved away, and the fractured surface that's left of the specimen is what is then replicated using the electron bombardment guns. After replication, the specimen is removed and the replica floated onto water or acid or base so as to dissolve away the specimen, because what we look at in the electron microscope is just the replica. What do we see by freeze etching? For example, in this electron micrograph of a freeze etched replica, we can see the nucleus with its pores, and out in the cytoplasm, endoplasmic reticulum. In this micrograph, at higher magnification, we can see a mitochondrium which has been cross-fractured, and inside the mitochondria we can see cristae with some of the fracture faces exposed. We can see these better on this vesicle. Here, in this vesicle that's protruding from the surrounding cytoplasm, we can see bumps. At first, we didn't understand what those bumps were, nor did we really understand what the fracture faces were. And the first indication of what was going on came by examining electron micrographs showing membranes that had been cross-fractured in one region and face-fractured in another region. It became clear when we looked at these that if this was a bit of membrane, what was happening was that the membrane was actually being fractured. A portion of it was being removed so we could see inside the membrane. Often we got a vast exposed region of the membrane. Now remember, what we look at by freeze etching is actually a replica which is formed of the fractured face. And on this replica, the sculpturing we see, these bumps, represent transmembrane proteins that are exposed by breaking the membrane, by fracturing the membrane in half. For example, in the red blood cell preparations we were working with before, if this line represents the fracture through a cell membrane or through a cell, what happens is that half of the membrane, as well as the surrounding material, is fractured away. And what we're left with over here is the half of the membrane closest to the protoplasm. The half membrane closest to the extracellular space has been fractured away. Whereas here, we fracture through the cell itself, scoop it away, and what we're looking at is the half of the membrane closest to the extracellular space. If there are any transmembrane proteins in these membranes, then they show up as bumps on this fracture face or on this fracture face. The ability to split membranes in half by freeze fracturing means that we can look at the internal components of a membrane. In other words, we can visualize directly some of the transmembrane proteins and thus relate the function of a membrane to some of its structural properties, something we'll be talking about at greater length in the next program.